Hi, I'm Scott, and you're watching my quickfire guide to cellular respiration in the human metabolism. In both aerobic and anaerobic respiration, the first stage is always glycolysis, which starts with taking our 6-carbon sugar glucose and phosphorylating it with hexokinase, leaving us with a molecule called glucose 6-phosphate. This is then rearranged by another enzyme known as phosphohexoisomerase into fructose 6-phosphate. From there, we phosphorylate the molecule yet further with phosphofructokinase 1 into fructose 1,6-biphosphate. Adelase then splits apart the molecule into both glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, but we can't use the latter, and so triose phosphate isomerase is used to convert it into the former. These two copies are then phosphorylated again via glyceraldehyde 3-phosphate hydrogenase into 1,3-biphosphoglycerate. The reaction for this uses an inorganic phosphate group and a molecule of nicotinamide adenine dinucleotide, or NAD for short, and produces a reduced NAD and proton per molecule. These are used later on. Next, phosphoglycerate kinase removes one phosphate group from each molecule, phosphorylating 2 ADP molecules into ATP and producing a couple of 3 phosphoglycerate molecules. Phosphoglycerate mutase then shifts the remaining phosphate group and converts our molecule into 2-phosphoglycerate. Enolase is then used to dehydrate the molecules to phosphoenol pyruvate. Then, at last, pyruvate kinase produces our 2 pyruvate molecules, phosphorylating another 2 ADP molecules in the process. In terms of energy, we don't actually get much production in this process, which is why it's pretty inefficient for powering the human body for long periods of time alone. Anaerobic respiration barely advances past the stage. An enzyme called lactate dehydrogenase converts our pyruvate to lactate, while oxidizing our reduced NAD once more so it can be recycled for more glycolysis, but other than that, this pathway has come to a standstill. Next, we have the oxidative decarboxylation of pyruvate. Until now, the processes we've seen have all occurred outside the mitochondria, within the cytoplasm. Now, pyruvate enters the mitochondria's outer membrane and pyruvate dehydrogenase converts it into acetyl coenzyme A and a carbon dioxide molecule, reducing a molecule of NAD in the process. We can now move on to the Krebs, citric acid, or tricarboxylic acid cycle, whichever you wish to call it. I'm going to go ahead and split this into 4 main sections based on the amount of carbon present at each section. In section 1, we see our 2-carbon acetyl coenzyme A joining with the 4-carbon end product of the previous cycle, oxaloacetate, via the enzyme citrate synthase. This unsurprisingly leaves us with citrate. The process involves the addition of a water molecule and the removal of our coenzyme A. Next, aconitase adjusts the molecule twice via dehydration and hydration, resulting in isocitrate. We can now move on to section 2, where isocitrate dehydrogenase catabolizes isocitrate into the 5-carbon molecule alpha-ketoglutaric acid and a carbon molecule, which is taken up by oxygen for removal. This process reduces another NAD molecule. Similarly, in section 3, we see our coenzyme A making a return and carrying out the same action when forming succinyl coenzyme A, a 4-carbon molecule. Now, we're in section 4, where quite a lot happens. First, succinyl coenzyme A synthetase converts our molecule to succinate. This process removes the coenzyme and phosphorylates a molecule of GDP, which in turn phosphorylates an ADP to ATP. Succinate is then converted to fumarate via succinate dehydrogenase, reducing 1-flavin adenine dinucleotide, or FAD, to FADH2. Fumarase is then used to produce malate via the addition of water, before at last utilizing malate dehydrogenase to reach where we started again at oxaloacetate, reducing a final NAD along the way. If you've not been keeping track, these processes leave us with 3 NADH molecules, 1 FADH2 molecule, and an ATP molecule, per pyruvate. And now we move on to the electron transport chain. The main source of our energy is generated here within the mitochondrial matrix. The process is a series of redox reactions at carriers that makes use of NADH and FADH2 molecules to pass electrons on between them, converting ADP and inorganic phosphate groups to ATP before depositing the electrons at an exogenous oxygen along with 2 protons to form a water molecule. The final results are shown here for one glucose molecule. Each NADH molecule results in the production of 2.5 ATP molecules, and each FADH2 molecule produces 1.5. Coupled with the 4 ATP molecules from the Krebs cycle and glycolysis results, we end up with a total of 30 molecules of ATP for each glucose molecule.